Greetings and welcome to the encore presentation of the Weinberg Group's March 2012 webinar entitled New Biosimilars Guidance. Has anything changed? All participants' lines will be on a listen-only mode throughout the call. However, if you'd like to submit a question or comment to the presenters, click the button marked Ask a Question on your event window and submit your question. All questions will be answered via email within a week of the presentation. To start, I would like to turn the conference over to Claire Robertson. Ms. Robertson, please go ahead.
Thank you. Good morning and good afternoon, everyone, and thank you for joining our webinar replay. The long-awaited FDA guidance regarding biosimilars has arrived, and those of us who have been patient and waiting for it are now looking for the impact of that guidance. My name is Claire Robertson from the Weinberg Group, and I will be moderating this encore presentation of our webinar titled New Biosimilars Guidance. Has anything changed? The Weinberg Group is a scientific and regulatory consulting firm serving pharma, biotech, and device clients. We are based in D.C. and also have an office in San Francisco. Our speakers today are Drs. Nick Fleischer and Bob Roth. Nick Fleischer is Vice President, Clinical Pharmacology and Biopharmaceutics for the Weinberg Group. He has 36 years' experience in biopharmaceutics, pharmacokinetics, pharmacy, and clinical pharmacology. His career includes several positions at FDA, the most recent of which was Director of the Division of Bioequivalence, Office of Generic Drugs, Center for Drug Evaluation and Research, CDER. Dr. Roth is Vice President and Worldwide Medical Director at the Weinberg Group. Bob provides consultation in all phases of research, development, and marketing of FDA-regulated drugs, biologics, and medical devices. Bob has provided tremendous support on projects concerning strategic issues, clinical evaluation, data management and analysis, development of product support documents, regulatory support, and even some litigation support. During the course of today's webinar replay, you will be able to submit questions, and we will try to answer as many as possible during our live Q&A session with Dr. Roth. However, we will be emailing all responses within a week of the presentation, along with a copy of the slides. With that, we will begin the replay.
And, I'd like to extend my welcome to everybody that's on this webinar also. As you see, the agenda for today's webinar will cover several topics. We will start with a brief overview, and the overview will cover issues as to why these topic is a very critical topic to be discussing today when this topic has been discussed many times before. And the next agenda item will be a discussion of the new FDA draft guidances and what these guidances teach us. We will also spend a little time talking about the quality considerations because these seem to be the essential component of these new draft guidances as we probably expected to be seen. So we will spend some time talking about why the quality considerations are so important to the biosimilar concept. And we will also talk about where we are currently in view of these new guidances, in view of where we've been regarding the biosimilars and biogeneric topic as it was formerly known. Dr. Roth and I and others at the Weinberg Group have published and have presented on this topic for many years now. The concept of biosimilars or biogenerics is not anything new for us. In fact, as I was preparing for this webinar, I went back and looked at some of the slides that I used in previous presentations, and it was a presentation that I gave back over 10 years ago on this topic, and my concluding slide in that presentation was the need to overcome the legal obstacles and the scientific issues if we move forward with generic biologics or biosimilars. As we now know, the legal obstacle has been overcome, and I will touch upon that a little bit later, but the passage of a recent act that amended the PHS Act to enable an abbreviated pathway for biosimilars certainly has done a lot for the overcoming the legal obstacle. And now that we have the guidances issued by the FDA, some of the scientific issues, which I alluded to back in 2001 as basically clarifying the characterization, is very well enunciated in these new guidances. However, we ought to note that the process here is a little bit different than what we've seen back with the 1984 Hatch-Waxman Act, the 1984 law amended the Food, Drug, and Cosmetic Act to introduce an abbreviated pathway for generic drugs, and basically amended the law. Following the law, there were implementing regulations that were created by the FDA as to how they would implement the law, and subsequent to that, we're seeing guidances coming out in how FDA then further implements their regulations. Right now, at the present time, we certainly haven't seen that kind of a scenario because the law has been passed and is in vogue. However, we have not seen any implementing regulations from the FDA, but we do see guidances, which is a little bit not the way it was for the 1984 Hatch-Waxman Act. The other thing that it was very instrumental back in spending a little time on the history of the bioequivalence aspects and generic drugs is that back in 1986, the Food and Drug Administration held a monumental hearing on the whole concept of bioequivalence. Now, interestingly enough, bioequivalence was not a difficult or really intense or complex science. However, the FDA felt that it needed to have a significant hearing to hear all views on what would be determinants of bioequivalence. And this hearing back in 1986, as I said, was a 3-day hearing. It was chaired by Commissioner Frank Young and Deputy Commissioner Norris. And as we've seen from that, the bioequivalence concept has evolved, and certainly since 1984, many generic drugs have been approved and have been accepted as being truly bioequivalent to their innovator products. Now, as we've seen for the people that have been involved in the generic world and in bioequivalence world, the 1984 Act brought upon a lot of litigation, and it's still ongoing today. And just to kind of summarize a summary statement from Dr. Woodcock, the Director for Center for Drugs, at a recent talk that she gave, that the new biologics law will certainly keep FDA and lawyers busy for the next 10 years, which I certainly believe will happen. So to go on to just an overview of why this is important, there certainly is a significant market for follow-on biologics or biosimilar products. As we know, many recombinant drugs are coming off patent, and by some estimates it said that about 75 biologic drugs will be losing their patent protection in the year 2012. Many of these products are based on cloning technology, which is a complex area, and as it will be noted later on, it will be relegated to only a few players in the field that have the capability to manufacture some of these products. And as we've seen, the cloning technology and the development of biosimilars has been a very important component of drug development for the past 20 years. Many of the newer therapeutic modalities that are out there are biosimilars relying on these technologies. Some of these drugs are blockbusters, and if we define them in terms of revenue. The dialysis and cancer market for erythropoietin garners up billions annually. As was stated in just today's New York Times article, Amgen, the manufacturer of several erythropoietin products, has garnered $40 billion in sales in the last 23 years. However, just yesterday, the FDA approved a new drug that is very similar to an erythropoietin, Amontis, but it's different enough that it avoids the patent issues that may be relegated to the comparison to erythropoietin. And it will be very interesting to note when we are able to see the summary base of approval for Amontis if it had somewhat of an abbreviated pathway, since it did have some similarity to the erythropoietins. The Enbrel, Remicade, and Humira, which I've listed on this slide, have been in the top 10 in global sales in the year 2010. And in Europe, the sales biologics make up 7 biologics make up the top 10 sales in the European market. As we know, as I said earlier, many of the products for like rheumatoid arthritis arthritis and Crohn's disease and other inflammatory disease rely on these new modalities for treatment. The changes that have occurred with the new act, the amendment to the Public Health Service Act, makes this a revenue opportunity for people that want to get into this field. Now, interestingly enough, as we sit here and we give this webinar and people listen to this webinar, down the street from our office in Washington, D.C., the Supreme Court is having hearings on the constitutionality of the new law. Now, whether the Patient Protection and Affordable Care Act, which is part of the Biologics Price Competition and Innovation Act, are somehow intertwined with the hearings that are going on before the Supreme Court will be very interesting, because if the Supreme Court finds those laws to be unconstitutional, we'll then be back at the starting block of not having legislation that would permit an abbreviated pathway for generic biosimilars or biosimilar products. Something to look for when the Supreme Court decision comes out, whether that will be impacted or not. What the change in this act did, it actually streamlined a pathway that would then reduce development costs and hopefully the price that people will be charged for some of these biosimilar products. As we know, the price of these drugs are extremely high, so even a small percentage in savings would be very large for people. While in the small chemical world, where a saving in generics may not be significant enough when you consider it percentage-wise, but when you're talking about a drug that may cost something like $40,000 a year, and you can save 10% on that, that is a substantial saving. So even though we're talking about small percentages, the actual savings could be significant to the consumers that have to take these kind of products. If we look at what has happened in the past and look back, it appears that the European authorities are certainly ahead of the United States in granting approval for some of these biosimilar products. We know that there are right now at least, there are 14 biosimilars that have received marketing authorization in the European market. Several of these are for the same product, but they have different trade names. In the EU, we know there are 2 somatotropins, 5 epoetins, and 7 filgrastim products that are marketed. As I said, the United States is a little bit behind on this, and we have 2 what would be considered FDA approvals of biosimilars, so-called biosimilar products for human growth hormone or somatropin, which are omnitropin, valtropin. But however, these products, the omnitropin and valtropin, were approved as drugs, and they are listed in the Orange Book, which certainly raises a question of, will the FDA have a similar listing available for products that are approved as biosimilars? By that I mean, will practitioners know what the products that are approved as biosimilars, and more importantly, when I get to the subject of interchangeability, whether these products have been designated as therapeutic substitutes by the agency and will be substituted. So I think one of the questions that remain unanswered is, how will the FDA address the issue of listing for these products and identifying products that are biosimilar? For right now, the growth hormones, the somatropins, the omnitropin and valtropin, while they are listed in the FDA's Orange Book, they are all listed as BX, indicating that they are not substitutable for the innovator product. Many years ago, and probably even currently now, we still hear the concerns raised by the innovator companies that manufacture some of these products, and whether these concerns are still valid or not. Many of these concerns were raised in citizen petitions, and they go back over a decade, and these were the scientific issues that were raised, and many of them revolved around the issue of, can a product manufactured by another process be considered the same? The whole sameness issue is a real germane topic here, and I think as Dr. Roth, Bob Roth, will discuss later on, the characterization issue becomes monumental in convincing the FDA that a biosimilar product is close enough to then be able to, for FDA to be able to decide what types of studies will be needed further on to get a product approved. So the sameness issues are issues that are specific for recombinant proteins. We have issues of how close is close. We know that for a small chemical, we know what close is, but how close does a biosimilar product have to be? And the big question is, can two independently manufactured proteins ever be truly identical? We also will need to have an assessment. The assessment will have to be technology dependent to show sameness. So there are a lot of issues in the analytical techniques that will be utilized to show sameness. And small differences can be as important as large differences. So even a small change can be very influential in both the toxicology and the efficacy of a biosimilar product. And what the FDA will have to kind of consider is predicting of the pharmacology versus the risk. We know that for small chemicals, they have to be shown to be pharmaceutically equivalent and bioequivalent. We've had instances where FDA has already crossed this bridge. Some have been favorable to the generic drug industry, and other cases it has not. Recently, FDA approved generic enoxaparin and deemed it to be both pharmaceutically equivalent and bioequivalent. As of now, we have not seen any generic forms of insulin, and the question is sameness of an issue with insulin? Certainly conjugated estrogens, that there's still no generic form of conjugated estrogens. Truly bioequivalent conjugated estrogens is definitely a sameness issue. And we also face this issue of benotropins, and there are numerous others where this issue of sameness has been in the forefront. But certainly for the biologics, it's going to be a very big issue to show the FDA that there is enough sameness between a biosimilar and the innovator product for then the FDA to make a decision on what other studies may be required. The active ingredient issues are very different for the biosimilar program than they are for the classic generics. In the classic generic world, we know that there are many sources of what would be considered identical APIs. We have sufficient testing methodology, and the testing methodology is relatively straightforward in the chemistry and manufacturing to know that an API manufactured by one company is highly similar or can be regarded as same from another manufacturer. In the biosimilar world, there are certainly technological barriers to API development. They are much greater, and there will be fewer sources for manufacturing of some of these APIs. There will be far fewer companies that have the resources to manufacture some of these, and it will certainly involve a different generic model that we're accustomed to for the generic drugs. In the generic drug world, we know as a standard model that a generic company would ordinarily identify a source of an API. They would then develop a formulation of that API into their own formulation that they think will be bioequivalent to the innovator product that they're trying to copy. And then they will hire a CRO, a contract research organization, to do the bioequivalence study to demonstrate bioequivalence of their formulation to the innovator formulation. It's very likely that the model for a biosimilar program is going to be totally different than the current generic model that we're accustomed to in the generic drug world. It's very difficult to match the biological contaminants, the protein refolding, and some of the processes and post-translational modifications that may occur in the manufacture of a biosimilar product. In addition, many of the APIs in recent years have been foreign sourced. Many come from India, China, and other parts of the world, and this may result in greater scrutiny. As we know, there has been an issue with heparin that was sourced from China several years ago, which required additional testing to be implemented, and certainly the FDA will be paying a lot more attention to foreign source material than they may have in the past. And importantly, as those that have spent time reading the recent guidances, FDA has clearly said that for a biosimilar there will not be any kind of a Type 2 drug master file as there are currently accepted for small chemicals in the generic drug world. This is very important, as I said earlier, because the generic model most often relied on a drug master file or an alternate source of the API, which demonstrated all the information that would be needed to know about the active pharmaceutical ingredient. The FDA has clarified some views after the Omnitrope situation. The FDA guidance that has appeared is based on or is very similar to the European guidance, with the big difference being that in Europe they've also developed drug-specific guidances, and Dr. Roth will be speaking about those later, or at least allude to those. The concept of highly similar for active ingredients is a sufficient standard for determination of sameness, and we've seen some of this if you review the FDA's position on the approval of enoxaparin, where they've actually used very similar language to what's being stated in the current guidances. The similarity can be established without a reference to the proprietary chemistry and manufacturing data of the innovator. This was a concept that Bio and the innovators of biologics were strongly in favor of, that you had to know how the innovator made the product in order to be shown to be similar, but according to the current guidance, it's not a necessity to do that. And also, the identical manufacturing process will not be required. Formulation differences could be allowed, but only if they will not impact the critical features, things like stability and immunogenicity. The current medical knowledge of potential risks may deem certain animal toxicology studies necessary and others unnecessary. As I stated before, the characterization will be essential to show that these products are the same as the innovator or very similar to, for FDA to decide what additional studies may be needed. The guidances call for extensive comparability testing with the innovator. Bob will discuss some of these specific issues regarding the scientific aspects. However, I just want to raise the point that when we do bioequivalence studies for small chemicals, it's not a difficult concept to be able to obtain the innovator product and to do the bioequivalence studies and even do the initial testing that is required to develop the formulation. However, when we're talking about the biosimilars, and if we follow the concept that is in the guidances, it will require a large amount of innovator supply to be able to do much of this testing, and it certainly will involve a large cost because many of these products are very expensive. Companies will have to ascertain obtaining several different lots of the innovator because the guidances call for that to show that there is heterogeneity in the product. So there will be that additional cost, which we don't see in the current market. What will be required for biosimilar applications? They have to use the same mechanism of action as the reference product. The conditions of use and the labeling will have to be previously approved for the reference product. The biosimilar must match the reference product by route of administration, dosage form, and strength. And the facility that it's manufactured is a given that will have to meet standards designed to assure that the biosimilar continues to be safe, pure, and potent. Now, the important concept that I think many generic companies are concerned about is the interchangeability. As we see in the Act itself, it is not a requirement for a biosimilar to be interchangeable. In fact, the first step in being interchangeable is you have to demonstrate that you are a biosimilar product and meet the requirements of biosimilarity, and then you go on and do the additional testing that FDA would require to show interchangeability. To prove interchangeability, as I said, the drug must be biosimilar to the reference product. And by biosimilar, it can be expected to produce the same clinical result as the reference product in any given patient. And the way that the FDA has, at least in the Act itself, has defined biosimilar is if the biosimilar is administered more than once to a patient, the risk in terms of safety or diminished efficacy of switching between the biosimilar and the reference product is not greater than the risk when the reference product is used without switching. The way to do this would be a multiple switch study, and as of this time, we do not know what exactly a multiple switch study would look like, as we've seen no guidance on that at all. Interesting concept in the guidance that we need to pay attention to is that there is some kind of a circular argument in the concept of requiring pediatric studies. As I said earlier, the interchangeable product ordinarily would not have to demonstrate that they have to meet the requirements of Pediatric Act and show pediatric studies or at least address the issue. However, an interchangeable product has to be biosimilar. A biosimilar product, by the FDA's guidance, would be a new active ingredient, and the new active ingredient needs to meet the pediatric requirements. So there's a little bit of a disconnect here in FDA's guidance about interchangeability and requirement for pediatrics. So what are the best practices in pursuing biosimilars? You have to define a regulatory strategy, and that's critical. It needs a detailed data package, and Bob may allude to some of that, or will allude to some of that in his presentation of what may be required. More importantly, that there will have to be high level of collaboration with the FDA. As the guidances state, the FDA will need to be provided with a complete, in-depth, comparative analytical characterization and in vitro data in order to get the best advice on what additional animal and human studies may be required. It will require communication often and more likely about everything, and the question is, will there be more pre-IND meetings and IND meetings than we see currently in the current development world? For the FDA to be able to participate in the development of biosimilars, they may want to see user fees earlier because certainly they will be spending a lot more time in advance in reviewing much of the analytical data that will be presented in characterizing the products and in meeting with sponsors that want to develop biosimilars. And it's very important that sponsors that are really going down this road to use precedents to their advantage. We envision that there will probably be longer review times for some of these products, so the question of whether a 10-month review that we see currently in the NDA world will be applicable is unknown at this time. And the question of whether the user fees that will be paid, will they be the same for a biosimilar as they would be for a current BLA, or will they somewhere in between what the generic drug user fees that are being contemplated? And a plan for scientific advantage about the approvability from the innovator is very important because there will be challenges, as there were back when the generic drugs first started being approved under the Hatch-Waxman Act. So the innovators will certainly start looking at scientific arguments as to why FDA should not be approving a biosimilar and, more importantly, an interchangeable product. So at this point, I'd like to turn it over to Dr. Bob Roth to continue the presentation.
Thank you very much, Nick. What I'd first like to do is just for context to help answer the question of what might be new post these guidances. I'd like to say just a few words about the biosimilar approvals to date. This slide shows briefly what kind of work went into the omnitrope and valtropin growth hormone development programs. These, of course, were done years ago. But as you can see, there was extensive chemistry work that was done on these. There was a lot of non-clinical test tube and animal work that went into pharmacodynamic responses. There was extensive animal toxicology that was done. There were standard bioequivalence PK studies that were done. It is noteworthy that if I recall right, it was valtropin that had a widened acceptance level for the confidence interval. It was a 70 to 143% confidence interval that was deemed acceptable. So not strictly bioequivalent by ANDA requirements, but close. There was a lot of additional human PD work that had to do with other growth factors, insulin-like growth factor and somatomedin C that are influenced by growth hormone administration, and then a lot of clinical work that was done. The omnitrope, for example, had a 9-month comparison efficacy safety study to genotropin, a non-inferiority study, and many other studies that had to do with when formulations were changed. Endpoints were typically a year or more, up to 15 months, looking at growth endpoints. Safety went out to 36 months. And altogether, there was about 200 patient years of exposure for each of these approvals. So as you can see, a very, very extensive development programs. Now, what we've learned from the guidances and how FDA will look at the next biosimilar that comes along after the growth hormones are started on this slide. First of all, there is no concept of a biogeneric. The concept now is biosimilar, and what was described over a decade ago in terms of the hope that someday there would be biogenerics, that is not on anyone's horizon. Abbreviated development programs have been very extensive. That was what was described in the previous slide, and in some respects, it may actually be more difficult to show comparable therapeutic profiles between a reference product and a biosimilar than it would be for a completely new biologics development program. It's highly unlikely that there will be any kind of substitutability process that will be defined in the near future. It's very clear that the FDA evaluations will be product-specific, and as Nick had mentioned earlier, it's expected that because of the high cost of these products and their development, that cost savings will be fairly modest. I'm not going to go through them, but there are a number of product-specific EU guidances and concept papers that help sponsors develop follow-on products in these areas. FDA does not, at this point, there's no indication that FDA will come up with something similar, at least in the near future. A few initial concepts about these guidances that just came out a month or so ago. There's 3 guidances. The first one is termed the scientific considerations, and this is a guidance that is fairly robust discussion about things like structural analyses and functional assays and animal data and clinical studies, and sort of goes into the whole range of things that go into developing a biosimilar. The second guidance is termed a quality considerations, CMC. It actually covers, there's quite a bit of crossover and covers some of the same issues, but it goes more in detail into discussions around expression systems and manufacturing processes, functional assays for potency and receptor binding studies and things like that, and gives some clarity into the sorts of understandings that FDA expects that sponsors will have for their products, and very specifically the comparison of their product to the reference product. The third quote-unquote guidance is really a question and answer series of very brief range of topics. I'm not going to go into these today, but these are things like whether the biosimilar needs to have or could be allowed to have a different formulation than a reference product. And these are all yes/no answers, and I won't go into them today. Now, as some general kind of first thoughts about these guidances, number one, FDA has given great flexibility. Like I said, there are no product-specific guidances like in the EU, and there really is no definition of what an appropriate data set would be. It's very clear that the biosimilar submissions that come in will be evaluated on a case-by-case basis. There is no one-size-fits-all. And a third basic message, I think it harps back to the slide that I showed with the very extensive development for the growth of the Valtropin and Omnitrope. The question has been really, since those approvals, once this new guidance comes out, is there some hope that the guidance will define or somehow describe a development process that would be either very abbreviated or identify aspects of development that would be clearly unnecessary, things that would take the development program, like for Valtropin and Omnitrope, that were sort of typical 505(b)(2) drug programs, and take the next biosimilar and move it at least somewhat closer to the development program of a generic. Certainly not that far, but someplace in between. And the guidances do talk about a number of aspects of the active ingredients and equivalents and things that make it very clear that the development is really not going to move very far towards that intermediate zone between an NDA and an ANDA. And in fact, aspects of the biosimilar, things like what do you have to do to market the drug. There will clearly need to be a professional sales and marketing team, for instance. You're not going to be able to sell the drug simply on the basis of cost and things like that. All of these issues are clearly pointing to the biosimilar being much more similar to an old-fashioned new drug. Some other issues, other considerations having to do with the quality, the CMC part. The guidance stresses extensive comparative testing of reference and biosimilar products. And it doesn't go into how similar the products have to be. It really goes into the processes involved in identifying the differences and then having a way to consider what the importance of these differences are. The guidance goes into quite a bit about talking about analytical testing, things that are very technically involved, such as knowing as much as possible about three-dimensional conformations and modifications. And clearly these sorts of analytical steps will require very, very high-level specific expertise and experience. And the real bottom line, I think, to the way FDA is going to look at these products is in the third bullet on the slide. Once the similarities and dissimilarities have been defined, the question is going to be what is the potential impact of these differences on the desired properties of safety, purity, and potency. And unfortunately, that's the part, how does one actually go from identifying differences to describing their impact? That part of the guidance is nebulous. It's clearly going to be case-by-case basis, and I think it's almost certainly going to be a matter of sponsor claims that FDA then sort of comes back and requires more information on. Some other conclusions that we get in looking at these guidances, the old concept that was raised over a decade ago about the process is the product for a biologic, that is clearly no longer an issue for FDA. You can have completely different microbial manufacturing processes of recombinant proteins, and that can be okay. There's a lot of discussion about process-related impurities coming from the cell systems, the manufacturing systems themselves, and processing steps. And it's interesting, nowhere in the guidance is there a discussion about upper limit threshold levels for impurities. Say, what percent of the mass can be an impurity, or how many milligrams a day would be allowable. There's nothing like that. It's clearly FDA is going to consider the clinical impact of the impurities, and it's much more than just a numerical concept. It's very clear that as for any given recombinant process and manufacturing system, there are going to be innumerable impurities, process-related products, and things like that that will be identified. FDA warns that if these entities are not already understood, that qualification work will be done, and there's a point made in the guidance that FDA figures that it's basically going to be easier to purify out such entities rather than qualify them whenever possible. Now, one point that I think is going to be a little problematic for biosimilar sponsors is the last point on the slide. FDA will know a lot about the existing reference products that the public won't know, the public and the new biosimilar sponsors. This will be proprietary information. They'll know information that came up in problems 2 years back with the reference product. They may have some very strong opinions about strengths and weaknesses of particular analytical methods that may be based on FDA knowledge, but not general knowledge. And this is going to be an interesting question. Things FDA knows that the sponsor simply cannot know, and how is that dealt with? Now, what FDA says to do basically is to identify all possible characterizations of your product and the reference product, and then go through and create some sort of a fingerprint-like algorithm of product attributes. It's not at all clear, at least to me, what this means to FDA, what it means to us, how one should do this, and whether or not there are some precedents to guide sponsors. So it's very clear that they're going to want a lot of data, and they're going to want it put together in some fashion that tells a story. How that's going to be done is very unclear at this point, and the guidances don't help. Another sort of take-home message that I got from looking at the quality considerations guidance is that in many cases, I think the amount of data that will need to be generated comparing your product and the reference product is going to be quite a bit more than the data that would be needed, for instance, if you're doing a comparability, if an old-fashioned reference manufacturer is doing a comparability study when they move from one manufacturing plant to another or make some minor changes in the manufacturing process. So a lot of data, one can't simply go on the experience with old-fashioned comparability exercises. Now, jumping from the quality considerations to the scientific considerations, I've identified on this slide a few of the sort of take-home general principle messages that I took from it. A couple of quotes that I put in here to address areas that I have a problem with. FDA says at each step the sponsor should evaluate the extent to which uncertainty exists between your product, the biosimilar product, and the reference product, and address how you're going to do something about that uncertainty, how you're going to interpret that uncertainty. It's pretty nebulous to us exactly how you go from A to B. That is, it may be possible to identify the differences, but how you're going to address them and how you're going to go about trying to explain away the unimportance of the differences is a major unknown at this point. And we think that although this is all based on scientific data, that art of the arguments will actually be just as important as the data themselves. It'll be a matter of creating discussion that will allow the agency reviewers to be able to go beyond the observed differences and hopefully conclude that the clinical impacts are either minimal or predictable. The amount of non-clinical toxicology that will be needed, this is now according to the guidance, is going to be very dependent on what differences one sees in the chemistry between the biosimilar and the reference product, and what and how many residual safety concerns one has after these differences are seen. So if there are very minor differences, and if you can make an argument that the safety profile really should not be very different, it may be possible to skip much of the non-clinical toxicology. Unfortunately, the guidance does not give enough detail to tell how one would do this, but it does raise the possibility that non-clinical tox might be, if not avoided, made a lot less onerous. The same thing with comparative toxicology is that even though it may be possible to show differences in a rat between a reference and a biosimilar product, how one goes about interpreting these differences in terms of human risk is a question, and I think this is part of where the art of the argument comes in. If a sponsor is able to make a data-driven statement that rats are not people, it may be possible to avoid or minimize the impact of differences that are seen in toxicology. Now, a key concept that comes up in the scientific considerations has having to do with what will be need to be done clinically. Is that the extent of clinical evaluation will depend on the extent of residual uncertainty about the differences between the biosimilar and the reference products. Unfortunately, the guidance doesn't provide any particularly great direction for evaluating this residual uncertainty, and again, this is where the art's going to come in. I think it's intuitive that when biosimilar applications go to the agency, there will be differences that will be identified, chemistry differences, maybe some animal toxicology differences identified between the biosimilar and the reference products. The sponsor will then make the statement that these are differences, but they will not prove clinically meaningful. How FDA is going to respond to these conclusions and how much information they're going to require to come to a conclusion is completely unknown. As I mentioned previously, and the last bullet here is that FDA will have undoubtedly proprietary knowledge about the reference product that may in fact influence how they look at differences that you've shown, and they will have an ability to do this in a way that the general public would not. The key concept for the clinical work that has to do with endpoints is that the FDA says that endpoints need to be clinically relevant and sensitive to detect meaningful differences. And the sensitive to detect meaningful differences is a very useful concept, because they then go on to give the example with anticoagulants that pharmacodynamic markers such as the INR, which is a scientific way of looking at the degree of anticoagulation, may in fact be a much more sensitive way to compare two products in a clinical trial than to look at true clinical endpoints like stroke or hemorrhage. Obviously, for the typical new drug application, FDA clinically is most interested in true clinical endpoints, and it can be a real battle to get acceptability of surrogate markers, of PD markers, but it's very clear that from the guidance that this will be perhaps even not only acceptable but a better approach for the clinical evaluations and that need to be done. Obviously, if pharmacodynamic endpoints are the sensitive and useful way to compare your product and a reference product, there can be this can have a major impact on cost for the clinical development part of the program. A couple last comments and then get to a conclusion slide. Post-market safety is discussed in a number of places. Immunogenicity is brought up over and over, and FDA warns that there may be considerable amount of post-market work that would have to be done to evaluate, in particular, immunogenicity, which is going to be quite difficult to get a handle on, at least the differences, again, between your product and the reference product, get a handle on that from, a 200-patient study, comparative study. I recently ran across a recent therapeutic protein approval, not a biosimilar, but an enzyme, pancrelipase, that required a 10-year observational safety study, and an observational study looking for viral antibodies to viral contaminants. Very, very extensive post-market approval requirements. I would envision that something similar may be required for the biosimilars that come along. Again, the one last thing about the scientific considerations, they do address a little bit interchangeability in a couple of places. It's not covered very well. We really know nothing more than what Nick had mentioned earlier, which is that some sort of switching study will need to be done to show that there's no impact of going back and forth between one product or another. But this is still a big unknown, and the guidance don't help a great deal. So finally, in conclusion, that the question that was posed in the title of the webinar is, now that we have these guidances, has anything changed? My take on this is that the previous framework that one had for biogenerics and biosimilars before the guidance, really the focus was when is similarity between the biosimilar and the reference close enough, and the concept that very minute differences that might have only theoretical impact might be enough to hold back the acceptability of a biosimilar product. Now with the guidances, it's very clear that whole concept is out the window. Considerable differences are potentially okay, even to the point that amino acids can be different between the reference product and the biosimilar. So considerable, not minor at all, differences may be perfectly acceptable. So to my way of thinking, what has changed now are the questions, and now the question is, when are the dissimilarities meaningful? Unfortunately, there's not a great deal of clarity on the answer to this, and it's not that clear to me whether FDA has answers either. And I think that the principles of the guidances will hold, but how it all shakes out in terms of a case-by-case FDA evaluation of a product that comes in at stores is really going to be a work in progress. And I think it will take several biosimilar approvals and all of us looking carefully at the summary basis of approval documents to truly understand what process FDA will take in their considerations. So I would like to stop here. Thank everyone for their attention, and Joel, if there are some questions, we'd be glad to address them.
Okay, great. Thank you. This concludes the presentation. We have a number of questions from participants. I'm not quite sure we will be able to get to all of them, but we will email the answers to all in a week. The first question for Dr. Roth is, the omnitropin-valtropin biosimilar growth hormone development programs were quite extensive. Could any of the development work be avoided based on the principles of the biosimilar guidances if development of a new growth hormone product is initiated?
Well, this is obviously a critical question for the field in general because it has huge cost and time implications. We don't really know enough about the chemical differences between the biosimilar products, based on what we know from the approval documents published by the FDA and prior to that by the EU when the products were accepted. But from what we know, the products were somehow close enough. Now that question, that issue of what's close enough from a chemical perspective is obviously completely based on the technology, and as the years go by and technology for characterizing in this case proteins improves, it's going to be easier and easier to find small differences. I think that if it can be shown that chemical differences are minor, and if you can make an argument that whatever these minor differences are would not likely prove to be clinically meaningful, it's very, very likely that a lot of the non-clinical tox and pharmacology work, could be avoidable. Now, the greatest savings for a biosimilar development program would be if it was not necessary to investigate human efficacy, somehow because the predictive value of the animal PD results were thought to be high enough. To make a case that on the basis of chemistry and animal PD that it's not necessary to do human efficacy studies, it would be very, very critical that we develop some sort of a data-driven argument based on perhaps previous investigations and other data in the literature, perhaps for how tightly one can link animal results to human efficacy. Now, human safety, I think, will continue for the near term at least need to be addressed, especially with regard to immunogenicity. However, if the only human work that's truly necessary is safety assessment and perhaps post-market immunogenicity, development programs could clearly be greatly simplified.
Okay, great. Thanks, Bob. The next question is, how should one go about crafting the argument that observed differences between biosimilar and reference products would not be expected to influence safety or efficacy?
Well, the FDA guidances indicate that you need to choose non-inferiority limits for these sorts of questions, and that these limits, they need to be scientifically justified. They need to be pre-specified. That is, you have to really have a pass-fail set of criteria that are pre-specified, and they also have to allow sufficient sensitivity to detect significant clinical differences between the products. This is a lot different than I think the way a lot of development work is done, which is to do the experiment, accentuate whatever positive results you get, and in effect do an experiment and then take the approach of we'll know it when we see it. I think that kind of approach is going to be very risky. I think it's really going to be necessary to have a very prospective pass-fail program that FDA agrees to in advance. Arguments will need to be crafted both from the literature and precedent product information as well. In many respects, I think when arguments are crafted that observed differences are not expected to have a meaningful impact on safety or efficacy, these sorts of arguments are going to look a lot more like what a typical citizen petition submitted to FDA would look like. We've had a lot of experience in providing, scientific support for citizen petitions, and I think that the sort of way the arguments are crafted and presented is going to be very similar. Now, the scientific justifications in citizen petitions, of course, are put out in the public domain. This would not be the case for a biosimilar drug program, but I think that the content of the argument is going to be very similar.
Okay, great. I think we have time for one more question, and it is, if strict bioequivalence does not need to be demonstrated, how similar do the innovator and biosimilar products need to be, and how might we measure those?
Well, that's a good question. We don't have an answer at this point based on the Valtropin experience, in which a widened confidence interval of 70 to 143% was allowable for the bioequivalence study. There's certainly the concept that a widened confidence interval that something less than strict bioequivalence from the generic concept would be acceptable. With proteins, of course, you've got you may have very short half-life of infused product, but the pharmacodynamic endpoint, the real way the protein works, may be considerably longer. So it's certainly possible that you could have a short pharmacokinetic half-life, and for a biosimilar protein, you may have dosing that's much longer than the half-life because of the extended PD. That is, maybe you give the drug daily or even weekly. And so it could be argued that strict bioequivalence is not needed because it does not predict efficacy or justify the chosen dosing regimen. So all that we know from FDA's stance at this point, based on their scientific considerations guidance, is that whatever PK parameters are chosen to support biosimilarity, they must be predefined.
Okay, great. Thank you, Dr. Roth. This concludes today's webinar replay. We would appreciate it if you could complete and submit the short survey. Have a great day.